Hello, I'm Prashant Govindarajan, a final year dual degree student at the Indian Institute of Technology, Madras. Welcome to this presentation on generating drug-like molecules from gene expression signatures using transformer model. As we know, drug discovery is a pharmacological process by which new medicinal compounds with desired biological properties are designed and approved. The process consists of multiple stages and generally takes several years to develop a single drug. The chemical space is large and discrete in nature, consisting of around 10 power 60 molecules, and this is a complex multi-objective optimization problem that requires a drug to satisfy multiple properties. Deep learning approaches are expected to play an instrumental role in accelerating this process. Gene expression signature, or GES, contains the relative expression levels of a group of genes in a cell that are differentially expressed. The signatures are usually induced by either introducing drug perturbations, gene knockouts, or even a disease. For training a transformer model, a subset of the Link cell 1000 dataset was used, in which a GES is typically a vector of values for 978 landmark genes. The cell lines chosen were MCF7 and VCAP, signatures with a duration of exposure of 24 hours and a dosage of 5 or 10 micromolar were filtered. This way, there were around 31,000 signatures corresponding to 20,000 compounds. This is the network architecture of a transformer model with modified encoder. We aim to pose the idea of producing drug-like molecules from a given gene expression signature as a machine translation problem, where the input language is the gene expression and the output language is the SMILE string of chemical molecules. But here, gene expression signature is not a string or a sequence, but a float vector of length 978. So the encoder part here is modified to produce low dimensional representations for the key and value entities from the input GES through feed-forward neural networks as shown here. The decoder processes the outputs from the encoder and the output embeddings, performs multi-head self and cross attention, and sequentially estimates softmax probabilities of SMILES characters. Here, the encoder part not only takes the gene expression as input, but also some random Gaussian noise. This is to make the algorithm generate a diverse set of molecules for any given gene expression signature. So these are the results we got after training the model on 80% of the data set. These plots are training loss and accuracy curves. The blue curve is for the training data and the red curve is for the validation data. We evaluated the performance on 20% validation data. For each signature, 100 compounds were generated in the form of SMILES representation by varying the noise vector. The first plot shows the distribution of unique and valid molecules across the validation signatures. On an average, more than half of them are unique and valid. The second plot shows the distribution of synthesizable compounds, and it was noticed that almost all of the unique valid compounds were easily synthesizable. This is characterized by a metric called synthetic accessibility score, and it has to be less than 4.5. Further, the similarity with the true compound was measured using Tanimoto similarity. The rightmost plot shows the distribution of the similarity between the true compound and one of those 100 compounds across all signatures. The mean similarity is 0.252. Here are a few examples of the best predictions of a model. The left column consists of compounds generated by the model. The next column are true compounds. Although these are different and new molecules, certain important structural features are correctly predicted by our model by also maintaining a reasonably high Tanimoto similarity. We then evaluated the trained model on 148 unlabeled knockout gene expression signatures corresponding to 10 genes to generate inhibitor molecules. These signatures are a result of knockout of one of those 10 genes. 100 molecules were generated for each signature and were evaluated based on validity, uniqueness, and synthesizability. The best generated inhibiting molecules for each gene was chosen based on the highest Tanimoto similarity with the already known gene-specific inhibitor molecules present in a database called ESCAPE. From the plots, we can observe that many of the molecules are valid, unique, and synthesizable. In the next three slides, the best generated inhibitors for all the 10 genes are shown. For the genes AKT1, AKT2, AURKB, and CTSK, it is observed that the generated molecules not only have a reasonable similarity score with a known inhibitor, but also shares certain structural features with it. Same is the case for EGFR, PIK3CA, HTAC1, and SMAD3. For the genes MTOR and TP53, the generated molecules were exactly the same as the known inhibitors. A few drawbacks: we did not take into account the variability in experimental factors that influence gene expression, like dosage, cell line, and the number of hours of exposure of the drug. We did not optimize the molecules to enforce any biological or pharmacokinetic properties. Hence, incorporating experimental factors and using reinforcement learning approaches for property-oriented generation are major future directions. Further, rather than using smile strings to represent molecules, we can represent them as molecular graphs, which offer better expressive power. To summarize, a novel transformer model with modified encoder which can generate diverse molecules that can induce a desired gene expression signature is proposed. This is posed as a machine translation problem, and the model sequentially generates the SMILES characters given a GES. To conclude, the applications of this study range from accelerating the early stages of drug discovery to aiding in drug repurposing. I wish to thank my professors and scholars at IIT Madras and the authors of a previous study who were very helpful in providing the codes and the data set of their paper. Thank you very much.